In this presentation, Dr. Walker, Professor and Research Director at Texas A&M University Agricultural Research and Extension Center in San Angelo, discusses different types of sore mouth in goats, methods of control and treatment, and how sore mouth affects goat management. The American Goat Federation, AGF, was organized as a non-profit national association in 2010 to represent the interests of all organizations and producers engaged in the production and marketing of goat milk, meat, fiber, pack goat, and grazing services across the United States.
Well, good morning. It's good to be here. And clearly sore mouth isn't a huge problem, or there'd be more people here, but that's okay, it's early. Just out of curiosity, how many people out there routinely vaccinate for sore mouth? That's what I thought. It's not a, national issue. It's a big issue in Texas. It started out because of actually the screwworm. If it wasn't for the screwworm and the sore mouth providing a avenue of entry, or for the eggs to be deposited, it probably wouldn't be that big of an issue there. But it is a huge issue in Texas. And what I want to talk about primarily today is some new forms of sore mouth that we're seeing. So we've been doing research on sore mouth in Texas for a long time. This publication was put out in 1936, and a lot of what we know today was discovered back then. The disease runs its course in 25 to 30 days, sometimes as few as 14 days. Immunity following vaccination is sufficient to protect against field exposure. What that means is you might be able to show reaction to a vaccine even after they've been vaccinated, but in terms of practical coming down with the disease and causing a problem, a one-time vaccination seems to be sufficient, at least with the live virus vaccine that we use. Lambs from vaccinated ewes do not have passive immunity as in some other diseases, and vaccination of lambs, of infected lambs, can lessen the severity. Okay, so I did a little literature search to just look at the numbers. I think it was 512 studies since 1937 on sore mouth, and as you can see, the number has been rapidly increasing over time. Some of that has to do with the with infections of the disease in people. Okay, so several of these studies, quite a few of them actually, are about people that get exposed through livestock or meat handling or wool processing to the disease. So not all sore mouth is the same. And so I'm going to kind of contrast what we typically call sore mouth, or ORF, with a new form which is severe persistent ORF, or also called malignant contagious eczema. So first off, the regular ORF, we see scabs on the lips and nostrils. With nursing lambs and stuff, yes, it caused problems, but it wasn't a severe problem. In the severe persistent ORF, we have severe scabs on lips, nostrils, ears, eyes, feet, vulva, really any place, and even internally on these animals. Traditionally, it was thought of as a sheep disease, but more recently, and particularly in boar goats, it's definitely a goat disease. The duration of the regular sore mouth is 1 to 4 weeks. And this the severe persistent ORF, I'm going to call it SPU, is up to 12 weeks, and if they live that long. And again, mortality was rare, but with this SPU sore mouth, we have mortality of 10% or higher. Okay, so it's a big problem. And that's just, if you're not familiar with the disease or the difference between the two, As you can see, typically, yes, you get a few lesions on the mouth. On nursing kids, it slows them down, but they get over it and they go on and do fine. These type don't survive. So there's two competing theories, though, as to what's the cause of this severe persistent ORF. One is that it's individual animal susceptibility, compromised immune systems, or the other is the virulence of the ORF virus strain. So I'm going to go through in the literature basically all the reports I could find of this severe sore mouth. The first one I found was reported in 1992. There were 47 suckling kids that had all been vaccinated with an autologous live vaccine. Okay, that's kind of interesting. In other words, they were vaccinated with the strain that was on on that property. 2 to 30 days after initiation of bottle feeding, which I think began at 2 weeks in this case, 62% of the kids contracted this severe sore mouth, and they had a 65% mortality of those that did. The surviving kids recovered in 3 weeks. And so the point of this, and I'm going to point this out in each of these cases, I think, is that it showed up after a stress was induced. They were weaned off the dough and put on a bottle. Next report that I found was 1997 in Scotland. This was in wether lambs. There were 550 5-month-old wether lambs that had been shipped. The truck broke down, so they were on the truck for 23 hours. There was a 0.4% death loss on the truck, and another 0.4 for a total of 1% death loss within the first week. So again, these lambs were stressed. 100 head developed the ORF 7 days after delivery. 20 head had the normal ORF and recovered in 2 weeks. And then 80 head developed the severe ORF, and the surviving lambs recovered in 4 weeks. 40 head died, a 50% mortality in that case. Again, we have stressed animals that were coming down with this. Some came down with the normal and were able to apparently resist it, but half of them, or over half, were not. In San Angelo, Texas in 2000, there were 220 boar goat kids. All had been vaccinated shortly after birth. 22 head developed the typical ORF, and 4 head developed the severe ORF. Sheep and goats on the same property did not develop this ORF. Now, there was no reason that these 4 head that developed the severe ORF, and they were eventually euthanized, so you could say 100% mortality there. They were all pretty well treated the same, and so there's no, but so there could have been an individual genetic difference in them. Yes, Bob?
The 224 kids that y'all vaccinated, those were vaccinated with what?
They actually would have been vaccinated with the goat vaccine. Yeah. A report in Israel in 2000 in Asaf's sheep that were not vaccinated. There were 4 different herds, and you can see that 4 to 6% in each one of these herds came down with this severe ORF. That's what it looked like in these sheep. And the authors of this study concluded that the Asaf breed is more sensitive to ORF than other breeds of sheep. So again, we have an indication that there might be some immune deficiency in either by breeds or caused by stress. A study was done in Italy in 2008 where they took 5 goats and 2 sheep that were unvaccinated and 3 weeks old, and they experimentally infected the inner thigh with the malignant contagious eczema. There was no difference between the sheep and the goats, and ORF virus did not produce any sort of malignant lesions. They suggested that it was host related. In other words, the animal was the cause when these severe outbreaks. However, I would say two things about this. One, vaccinating on the thigh may not produce that, because the reason we vaccinate on the thigh with a live virus is that's not where the disease normally causes problems, and by vaccinating there, you may not have that problem. Which brings up a point real quick. Who knows why we recommend, and not everybody does this, some people vaccinate in the ear, but we always vaccinate, in an atypical place for the disease to multiply and spread. Why did they suggest vaccinating on the inner thigh? Does anybody know? Well, the theory was that we're using a live vaccine. We don't want it to get on the lips and nose of these animals where it normally causes problems. And they figured that the inner thigh of a lamb was the least likely place for another lamb to stick its nose and get that infection and actually cause the disease. In Turkey in 2010, there was 150 hair goats. 90 kids came down with typical lesions, recovered in 2 to 3 weeks, but 12 twins developed this SPU and had a prolonged recovery of 3 months. And they suggested that the increased pathogenesis was caused by an immune deficiency in these twin lambs. It was only twin lambs that came down with it. So again, some indication it's due to immunodeficiency in the individuals. We did another study in 2012 so this group was vaccinated with our standard ORF vaccine, okay? And this group was vaccinated with our severe ORF vaccine. And what you see is that and then they were challenged with either the vaccine they were vaccinated with or, in each case. So when we challenged the ORF vaccine with the ORF vaccine, we still had 40% that showed some reaction, but that reaction was very mild. It could have been just from the scarification of the skin, quite frankly. However, when we vaccinated them with this SPU, with this severe vaccine, they had a larger reaction, and more of them reacted. But just the opposite happened when we looked at this severe vaccine. If we vaccinated with it, challenged with the standard ORF vaccine, we had a greater reaction and a much larger scab formation. So bottom line is we know there's various strains of this ORF virus, and we know vaccinating with one does not give as good of immunity as vaccinating with the one that they're going to get exposed to. However, there's no indication that the virus that has been found in animals that came down with the severe form of this disease was any more protective against another strain. So is there any evidence that this SPU is related to the viral strain of the vaccine? Well, again, what we know is there's a lot of strains. A lot of them been categorized. There are different strains we find in goats and we find in sheep, but frankly, there hasn't been much indication that there's one or several different viral strains that are particularly virulent as compared to the others, and which is what we basically saw in this instance.
So those different strands like that, do they mutate, or is that the ones you found, is that all there is?
So when they do cell culture and they pass them through cell culture multiple times, they do start mutating. So I assume in the animal they will mutate as well. But to be honest, I'm not sure whether in vivo that happens. It probably does. We know when we pass it through cell culture that they mutate. So I would argue that the severe persistent ORF appears to be caused by individual immunodeficiency that results either from environmental stress or a genetic factor within the animal. So let's talk a little bit about vaccine types. All of the vaccines in the U.S. right now are a scab vaccine. Basically, you scratch young lambs, you wait 14 days, you collect that scab, and you produce a vaccine with it. It's a high potency vaccine. There's the other type of vaccine is a cell culture vaccine. The advantages of it is it's easy to produce and there's no contamination. There's a lot of concern amongst people that when we vaccinate with this live virus, that we could actually introduce a disease by doing such, another disease. And and the ease of production again would be a good reason. The disadvantages of the scab is it's labor intensive, and interestingly, in the last couple of years, for reasons we haven't figured out yet, scab production has not been near as great as it has been traditionally. So where we might be able to get enough scab to produce the vaccine that we sell by scratching, say, 100 lambs or so, we're now doing 200 and 300, 400 lambs trying to get enough scab. Many of them just aren't producing a scab. It could be that a lot of the lambs we're scratching we don't get to until they're a month or so old and they've already been exposed to the virus, and so they just aren't reacting to the vaccine when we scratch them. But that's not always the case either. Anyway, it's become an issue, quite frankly, in producing enough scab to produce the vaccine. Again, the disadvantage is the potential to transmit other diseases, but I would say in 80 years of producing the vaccine with millions of doses, millions of lambs vaccinated, there's never been a report of transmission of any other disease through this method. The disadvantage of the cell culture vaccine is low potency. It really doesn't work very well. The suggestion is to vaccinate annually with the cell culture vaccine. It is produced in England, and I think other places in Europe they use a cell culture vaccine. But again, whereas with our vaccine, the live vaccine, we normally we just recommend one vaccination at lambs, and that protects them for the rest of their life. A study was done in England that looked at the cell culture vaccine efficacy, and if you look at that, so this group of they had I think 730 farms that responded to this survey, and this group, the producers vaccinated annually for both the ewes and the lambs. This group just vaccinated lambs, and this group did not vaccinate at all, okay? And so you can see that although the 23% of the animals that were not vaccinated, of the flocks, I should say, these aren't animals, these are percentages of flocks, had animals that developed the disease. They did not report what percentage of the animals, but they did have animals that developed the disease. The ones that only vaccinated the lambs actually had fewer animals fewer flocks that came down with the disease, but that probably wasn't really significant. The point being that with that cell culture vaccine, the percentage of breaks is fairly high. So what should you do if you're in an area and you're dealing with this severe form of the sore mouth? Well, first off, Texas Vet, and I might say that right now there's in the U.S., two licensed vaccines, one that's produced by Texas A&M AgriLife Research and the other by Colorado Serum. They're both scab vaccines, and they both have a sheep seed that they use, okay? There is no vaccine that is licensed and commercially available that uses a goat seed as the source of the virus in there. Texas Vet Lab is applying, and hopefully we'll have a goat vaccine out in the next, within a year, I think. They're moving right along on this, so that's good. But the other thing is to produce your own autologous vaccine. And this is, to me, a little interesting because several years ago, I suggested that we ought to put out a brochure and tell people how to make their own vaccine. And I was told, no, you can't do that. You have liability if you tell them how to do something and they get in trouble and all this. But this was published in 2011. And how to make your own vaccine is in this publication, so I guess it's okay for me to tell you how to do it too. And it's basically you collect some scab and you mix it with an extender, and which is typically a normal saline solution. You can add a little bit of antibiotic if you want to protect against any bacterial infection you may get from the scratching, and you have a vaccine. Interestingly enough, again, back 80 years ago, the same thing was said in one of the publications that came out from the Texas Agriculture Experiment Station. And so if I had issues was afraid of having outbreaks of this severe sore mouth, I would collect scabs from my own animals and I'd develop my own vaccine. So how do you make a normal saline solution? A half teaspoon of salt in a cup of water. It's pretty simple, right? So I would take a cup of water, I'd put a half teaspoon of salt in there, I'd collect some scab, throw it all in the blender for a minute, and I'd go vaccinate my animals. If I had a flock, and I may have already even vaccinated, and I had a break with this severe sore mouth, what I would do is get that animal, create this vaccine, I'd revaccinate everything with it. I also would say, what were the stress factors that were happening that might have caused this? You know, do I need to feed more? Something like this. Did we have, some really bad weather a couple weeks ago? And so I tried to take care of both the environmental stresses. I would revaccinate, and I would keep a record if I knew which kids were coming out of which does. I would try to keep track of that, and maybe there's lines of does that also are more susceptible. Treatment-wise, so first off, if you're a commercial goat producer, you're not going to do much treatment other than, again, as I said, vaccinate. But treating individual animals probably is not worth the effort. If you're producing show goats, that's a different story. But levamisole, the wormer sold as Prohibit or Levamed, has been proposed as an immunostimulant. Not, as far as I know, there's not much data to suggest that really helps, but it's been suggested that could help. Again, vaccination with an autologous vaccine could lessen the severity. And then there's a few topical treatments. There has been some reports where these have actually worked. These are basically antiviral drugs that you can use topically. However, I have talked with our local veterinarian who has treated animals with these, some of these antiviral medicines, and he said he has not had much luck. But there has, excuse me, there have been reports of it being effective. So, any questions? Yes.
You can get an autogenous vaccine made by a professional lab.
Yes.
What's the difference between the way they would do it and this plan to take the scabs, put it in saline, and put it in your blender at home?
The question was, so, and I need to make a clarification here, and I probably won't get, I'm not very good with medical terms, so if somebody wants to correct me. There's an autologous, which is I've been talking about, and that's done for your flock on your property. And autonomous is done through a third party, and which is what you're talking about. And it can be used on your flock plus neighboring flocks that is thought to have the same. So you have to be licensed and that sort of thing to do the autonomous one, okay? But the question is, what's the difference between doing it yourself and getting a veterinarian that's licensed to produce these vaccines to do it for you? There'd be no difference. It should be the same, yeah. It's just, the only thing I would argue is, this is pretty simple, so I would just do it myself, but yeah. Bob?
No, when I mix my scabs and my saline solution such as that, blend it and put it in my bottle, how long in the refrigerator will that last before I need to do it again?
Don't really know the answer to that question, but I would guess a week or so. And you can keep, the other thing you can do is you can save vaccine, keep it dry. And the reason I say that is, so if the vaccine is kept cool and dry, if the scab is kept cool and dry, we know that can last for years actually. So, and particularly if you have one of these severe cases, you've got off of one animal more than enough scab to do, a lot of vaccines. So if I had that happen to me, I would keep some. I would just let it air dry and then save it and then make up a fresh batch. I mean, we know it persists in the environment for years. It's been definitely demonstrated to, I think, survive in dry areas for 15, 20 years. However, in England, where it's wet and cold and damp, it doesn't survive lots of times doesn't survive a year. So it does depend on the conditions. The comment was that he's had vaccine that actually survived for a year, just stuck up in the rafters of his barn. And that, I don't doubt that, because we know it can survive a long time, but we also know in certain conditions it doesn't survive. So the question is there a good time to harvest the scab? We harvest scab at about 14 days. And the reason we do that is that's about the time the scab's going to fall off. So it's easy to remove, it's less traumatic to the animal to pull it off. If I had this severe sore mouth, I would just euthanize, quite frankly, I'd euthanize the animal and collect the scab, because chances of him making it aren't that great in the first place, and even if he does, you might not have much, of an animal. And let's get started on trying to take care of the problem. Can sore mouth, and it doesn't matter whether it's the SPU or the normal, whatever it is, can be transmitted to people. There's lots of there's just lots of reports of it. So you want to be in fact, when you're vaccinating, you need to be careful, because you can cause the disease in yourself if you're not careful. It's recommended that you wear gloves and normal safety precautions. The statement was that the general consensus is, if you've never had it, you do not vaccinate, and if you do have it, you have to always vaccinate, and that's correct. Where we had Angora goats on the same property as the Boer goats that came down with this severe ORF, where they run together basically, and no, they weren't. They were in separate places, but I would say that where they were run had goats on it that had, this actually happened at the Sonora station, where the original sore mouth work was done. So I'll guarantee you every square inch of that property has had animals with sore mouth on it for 100 years. So the comment was that he's seen one case of the severe sore mouth, and it was in a Kiko herd. And I'm going to tell you, I have seen the severe sore mouth in an Angora goat too. I have a picture I could show you, but I didn't put it in here. But that's a good point. And the question is, in my mind, is it even in these boar goats? Were there some that were stressed? I mean, animals. You can have a big flock of animals. They don't all have the same environment, really, you know. As that one study that was done where it was only the twins came down. Maybe they were nutritionally stressed. Yeah, so I certainly don't think it's just a boar goat problem. And as we saw in a lot of these other studies, it wasn't just goats. It was sheep and goats, and yeah.
With the severe case especially, because of the mortality, because of the existing vaccines that are commercially available aren't really for you. What would be the recommendations if somebody has a case? How should they treat it, quarantine it? What should they do to try to prevent spread to other animals in their flock or herd? What would be those recommendations?
So the recommendation was if an animal comes down with the severe sore mouth, you know, what do you do? And certainly quarantine is always, you know, a good idea. I mean, we should do that. And I think the other thing, because I think it's there's a lot of evidence that it's stress related. And the one stress that we can somewhat readily ameliorate is nutrition. So I would up the feed on the rest of them, you know. I don't know that would do something, but that would be a reasonable thing that somebody could do, that maybe we've got some animals out there nutritionally stressed. Some of them are breaking with this severe sore mouth. Let's quarantine the ones that are and up the feed on the ones that aren't. Yeah, Bob. Okay, so the comment was that he hadn't seen any environmental condition that he can pinpoint causing the problem, but he believes it's more the genetics of the animals are all the same. And my only question would be, when you have these breaks, do all of the animals, clearly all the animals don't come down with it, or you wouldn't be in the goat business anymore. What percent?
Pretty high percent.
Of the severe sore mouth? So what that suggests to me we need to do, and maybe we need to talk about this later, is to try to do a GWA analysis on animals that break and animals that don't, to see if we can find a marker that might indicate which animals are susceptible. Does that make sense? Yeah. I don't know. Can you answer that, Steve?
Okay, so the question was, what's the cost of doing GWAS, and it depends. If it depends on what we call the genetic architecture of the trait, and so specifically, is it one gene? Is it a situation like Scrapie where one gene will do it? In that case, you're looking at a pretty small study, and you could be looking at $10,000 maybe. If the genetic architecture is, it's kind of in there's a lot of different genes and they each have a relatively small contribution, then what you need is a lot more animals to be able to slice that out. And so that one could get into to six figures pretty quick. Is that a reasonable answer to? It is, but you might do some kind of a seed situation where you know there was a little bit of research and you put in a little bit of money to just see kind of how did the initial data look. The big thing anyway, so we do the GWAS relatively routinely, and the one thing is is trying to get the samples where you know what happened and you've got the records, because usually the problem we get into is that being able to get DNAs, and we say, okay, this animal definitely was exposed but didn't get it, and this animal definitely got the severe form, for example. It's getting enough of both types to be able to do a proper comparison that's usually our problem. Money's an issue, but often the bigger issue is the samples and the records.
How many samples are you talking
Okay, so if it's a single gene thing, you probably can do it with 10 or 20 of each. If it's if it gets to be really complicated and lots of different genes, then you need. It's tough to lay it out, but you probably are, at least 50 affecteds, and usually with these situations it's a lot easier to get a healthy control than it is the disease animal. And so if you had a few hundred clean animals and then 50 in the smallest group. Does that help?
Yeah, great, thank you. So, question, if we got blood cards and gave them to producers that are having the problem and said, okay, collect a blood sample on this card are the ones that have the severe ORF, and ones that don't on the other card, that's all you really need to know, right? And we collected a bunch of those up, we could start the process. Okay. So we may need to talk about a study like that. Yeah. Anything else? Well, thank you. Good questions.
The American Goat Federation, AGF, is a nonprofit national association that represents the interests of more than 200 organizations and thousands of producers engaged in the sustainable production and marketing of goat milk, meat, fiber, and grazing services across the United States. This video was produced as part of the educational services of AGF. AGF also supports research projects that will benefit the goat industry by writing letters to accompany grant requests and providing information when requested. The board of directors is made up of representatives of member organizations, individual producers, and industry specialists from all parts of the United States. Director candidates are nominated and serve 3-year terms on the board. AGF is headquartered in West Lafayette, Indiana, and open from 11 a.m. to 4 p.m. Eastern Time, Monday through Friday. Visit our website, AmericanGoatFederation.org, where you will find information about goat management, healthcare, and other resources. While you're on the website, become a member of AGF and help us provide information to goat owners and represent all goat producers by providing information about our concerns and industry needs to government agencies, USDA, and researchers.